Hello and welcome to the Huxley Speaker Series once again. So our guest today, Dr. Manuel Montano, Assistant Professor at Huxley College of the Environment, where he's part of the Environmental Sciences Department. Dr. Montano received his PhD from the Colorado School of Mines in Applied Chemistry, where he was working with engineered nanoparticles in natural systems. He joined Western Washington University in 2019. At Huxley here, he's a part of the Institute of Environmental Toxicology and Chemistry, and his research focuses on nanogeochemistry, microplastics, and emerging contaminants. So a bunch of big words about very small things that I'm looking forward to learning more about. Please join me in welcoming Dr. Manuel.
All right. Yeah, welcome everyone. Let me just share my screen here real quick. And there we go. So thank you for that introduction, Stefan. As mentioned, my name is Manuel Montano. Again, I'm a professor here at the Western Washington University. Grew up in Colorado in a small mining town called Idaho Springs. And then I ended up going to college in Golden, Colorado. Did both my bachelor's and my PhD at the Colorado School of Mines, getting a degree in applied chemistry, where my thesis was mostly centered around the detection and characterization of engineered nanomaterials in environmental systems. And then I went to Duke for a couple years for a postdoc, looking at the interplay between more conventional contaminants like endocrine disruptors and that of engineered nanomaterials. And then I spent about a year and a half at the University of Vienna doing another postdoc, where I extended the work of my PhD to start looking at the natural nanogeochemical environment using all the tools that I had developed for engineered nanomaterial detection and characterization. And so now I'm here and I still study really small things. It just kind of changes day to day what those small things are. And so for the talk today, I'm going to give you a little bit of introduction into why we really care about this nanoscale and some of the challenges associated with trying to better understand these systems. And so just for an example, we'll walk through kind of the importance of nano, and I'm using a little cartoon here of these needles. You can imagine we have these really small things in the environment, but trying to find them is like trying to find a needle in a haystack. There's a lot of things out there, and these nanomaterials are at very low concentrations. And so we'll get into how we can go about detecting and characterizing these materials in the environment. And then beyond just looking at for these really small things in the environment, we want to really understand the environment as a whole. So not just the needles, but also all the little individual hay fibers that make up that haystack to gain a really holistic understanding of all the processes that are going on. And so before we really get into it, it's important to define what a nanomaterial is. Operationally, it's defined as a material having size dimensions between 1 to 100 nanometers. So that can be a particle where all the dimensions are under 100 nanometers. It can be a film, or it can even be a rod. And so we have a number of different important processes are driven by these very small materials, particularly in the environment. The nanoscale is important for driving things like catalysis or the transport of contaminants, and also various dissolution processes. And the reason why this nanoscale is so important is because as we decrease the size of our materials, we increase the specific surface area. So all chemical reactions happen at the surface. Atoms within the inside of a material aren't capable of reacting. All reactions happen on the surface, where the atoms of the material can actually interact with something else. So as we decrease the size, we increase the proportion of atoms on the outside, and therefore we increase the reactivity. And so as a consequence, we've been exploiting this capability of nanotechnology for several decades now, leading to the emergence of a field of engineered nanotechnology. But it should be noted that even though we're now starting to build nanomaterials ourselves, nanomaterials have always existed in the environment. And so here we have a couple of examples where we have these ferrihydride particles, which are responsible for that orange coloration you might see in acid mine drainage. And you can see the scale bar here is only 20 nanometers. So these are very, very small particles that are capable of being transported in environmental systems. And then if we look here, here's an example of an engineered particle. This is mesoporous silica. And again, just examine the scale bar here. This is 10 nanometers. And if we remember what the size of an atom is, it's only about a tenth of a nanometer, so on the angstrom scale. And so we're really looking at individual atoms within this electron microscope image here. So what are the challenges associated with finding these materials in the environment? Well, 1, small size, 2, low concentrations, and then 3, the fact that there's already nanomaterials present in the environment. As far as the kind of processes that these nanomaterials can participate in, they can interact with everything from binding to sediment and interacting with benthic organisms, from dissolving and potentially reprecipitating to generating reactive oxygen species, to absorbing contaminants, both organic and inorganic, metals like lead and metalloids like arsenic. They can be uptaken by biota. They can interact with the extracellular polymeric substances that biota might excrete. And then they can bind with other particles already present in the environment. And so we can have homoaggregation and heteroaggregation occurring. And so if we look at the sources of nanomaterials into the environment, and we kind of break our nanomaterials down into 3 broad classes, we have our engineered nanomaterials. So these are man-made nanomaterials that we intend to use for their nano function. And how they might end up in the environment is either through the use and disposal or the agricultural practices. So there's nanopesticides and nanofertilizers. So we might have runoff from the applied area. And so we can have the introduction of engineered nanomaterials that way. Incidental nanomaterials are also man-made, but they're not intentionally produced. So this would be things like tire wear or smokestack emissions or diesel exhaust. And so we can get entry into the environment through urban dust, through vehicle emissions, through stormwater runoff, and then waste and disposal. But the concentrations of all these materials really pale in comparison to the abundance of naturally occurring nanomaterials that are already present. And this comes from atmospheric deposition, from neoformation, where microbes can take metals in the environment and form nanomaterials out of them. We have sea spray generating a lot of salt crystalline nanomaterials. And then by far the biggest source is just going to be the erosion of rocky material. So taking our big boulders and grinding them down into sand and clay and breaking them down further into nanoscale materials. And so with this whole soup of nanoparticles out there in the environment, we really have to ask ourselves, what's important? What are we interested in studying? And really it comes down to 3 big parameters is how I see it. The most important parameter when it comes to nanomaterials might be size, because size is going to determine reactivity. And as a consequence, it might determine things like toxicity and how it interacts with other substances in the environment. On the concentration side, mass concentration is useful, but also number concentration is extremely useful because it gives us an idea of the distribution of the sizes of particles in the environment. And then composition, obviously, is going to be important. What is the material made out of? And so if we're looking at our soup of nanomaterials here, our engineered, our incidental, and our naturally occurring, how can we delineate between these different classes of nanomaterials? How can we tell the difference between all of these? Well, one way we might try is through the size distribution. So engineered nanomaterials are typically made with very narrow size distributions. So we want them to have a particular function, and therefore we synthesize them to have a particular size range, as opposed to the naturally occurring nanomaterials, which are again generated from rocky erosion and different processes. Those are going to be a little bit more polydispersed and have a range of sizes. However, when we enter into the environment, these nanomaterials are going to start aggregating and clumping together and settling out. And so the size distribution is going to change dramatically. And so it's really not going to be a good metric for how we can tell the difference between these different particle types. We can maybe try to use exotic features. So a lot of these engineered nanomaterials are made with very complex surface designs, Maybe they have specific kind of morphologies to imbue a certain kind of property. But again, when they enter into the environment, it's likely that they're going to lose a lot of these different properties, a lot of these features, and therefore we're not going to really be able to use them as a way to tell the difference between an engineered and a naturally occurring one. So really the most recalcitrant property that we have available to us is that of elemental composition. So when we make a cerium dioxide nanoparticle, for instance, we typically only make that to be cerium dioxide, as opposed to a clay mineral, which is going to be a whole hodgepodge of different elements that comprise that particular mineral phase. And so here we can see the difference. This is a mass spectrum. So it's showing the mass charge ratio against some kind of intensity. We can see for an engineered nanoparticle, we really only have one kind of element present. But if we look at a natural nanoparticle, we not only have cerium, but we also have lanthanum and a whole host of other rare earth metals that are present. Why this might be important is, well, if we think about the distribution of cerium and lanthanum in the environment, if we look at just a collection of surface waters, it really conforms to this really tight distribution of concentrations. And so cerium and lanthanum tend to exist with a specific elemental ratio of about 1.7 to 1 in the environment. And so any additional cerium from the engineered side is going to shift that ratio. And so using elemental composition, we can maybe tease out if there's additional engineered cerium present in the environment. And we can use that to contrast with our natural nanoparticles, which again have this somewhat consistent ratio of these different elements. Unfortunately, there's not a lot of good techniques out there capable of looking at the really small sizes. We tend to think of electron microscopy being the gold standard, and it is great. But if we try to compare these three particles, these are all titanium dioxide. One of these is engineered, one of them is naturally occurring, one of them is incidental. It's going to be really hard to tell the difference between these three, just trying to assign them based off of electron microscopy. So we need a little bit more sensitive method to delineate between these particle populations. Now, when it comes to quantifying composition, conventionally this was done by filtering your sample through a kind of filter, dissolving what was filtered out, and then you could get the composition and mass concentration. However, this approach really loses a lot of the interesting information that we're after. Remember, we're after things like size, we're after things like number. And so we need a new method that can get us that information while still retaining things like the composition of the material. And so that was what I did my PhD in, was helping develop this technique called single particle ICP-MS, where now we can start looking at individual particles and their composition. We can count up these particles as a way to not only get their composition, but also their size and their number concentration. And so how this works, it's easiest to explain it by going over how a normal ICP-MS works. So ICP-MS is inductively coupled plasma, mass spectrometry. It's essentially an elemental analysis tool. And so what happens is you aspirate your liquid sample into a plasma. And this plasma is running about 10,000 Kelvin. And so everything that goes in here gets atomized, it gets ionized, and you get this generation of ions that then goes through this instrument. And you have this mass filter that is going to basically gatekeep and only allow ions that you want to go through. And so in conventional analysis, we would have our dissolved sample. So each of these droplets would contain some amount of ions. And so when we sent them through the plasma, we get this constant stream of ions going through. And so you get this somewhat constant signal. And so you would average this signal and then compare it to a calibration curve, and you could get concentration out of it. Single particle ICP-MS, on the other hand, you don't dissolve your sample up at all. And so what you instead have is a bunch of empty droplets, and occasionally you have a droplet that contains a nanoparticle. When this nanoparticle goes through the plasma, you generate this big plume of ions all at once. And so this ion cloud is going to travel through this instrument now, striking the detector. And so if we look at the signal intensity with time, we're going to have a whole lot of nothing because we just have a bunch of these blank droplets going through. And then occasionally we'll have a nanoparticle go through where we'll have this big spike in intensity. And the thing about this spike in intensity is we can directly relate it to the mass of ions that hit the detector. And from that mass, we can calculate a size if we assume something about the geometry and the density of the material. So how could we use this in an environmental setting? So I'm going to give you a case study to show how this might be used. And so here we have Clear Creek in Golden, Colorado, which is where I went to school. And what we did is during Labor Day weekend, we sampled a site upstream from where people were playing in the stream and downstream from where they were tubing and kayaking and what have you. And what we were looking for here was actually the release of titanium dioxide. So titanium dioxide nanomaterials are used a lot in mineral sunscreens. And so by examining these two different areas, we were trying to see if we could determine the amount of engineered titanium dioxide released into the environment. So if we look at the downstream site, we can see all these spikes of titanium being generated. And so we think, wow, that's pretty cool. We're seeing some engineered titanium being released. However, we also see the same thing in the upstream site. And this goes back to that problem where we have natural nanomaterials present in the environment in addition to the engineered nanomaterials. So it's really hard to tease out the differences between these different particle populations. If we look at some of the other data, we can see, sure enough, that we saw spikes in the oxybenzone concentration, which is a component of organic sunscreens. And we saw them at the time of day where we would see people playing in the stream. And so you can see we get these spikes in the organic sunscreen component. We see spikes in the titanium as well, above that of the upstream concentrations. But if we try to compare to some elemental ratios of titanium and aluminum, and titanium and iron, some other clay mineral elements we might expect to find, we really can't tease out those differences that well. And so it really becomes a question of how can we start delineating between these engineered and naturally occurring particle populations. And so that's when I also worked to develop what was a fast switching quadrupole, where now instead of just allowing one element through, we switch the quadrupole really fast to allow at least two elements through within a given particle event. And so what that looks like is we have this multi-element particle, and we have the ions of both the silver and the red color here flow through the instrument. And when we're analyzing the gray, it's kicking out the red. When we're analyzing the red, it's kicking out the gray. And so what that signal looks like is like this, where we have our nanoparticle event. And if we look at kind of how that signal is distributed, sometimes we're analyzing the lanthanum, and then there's a settling time where we have to give the quadrupole time to switch over. And then you can analyze the cerium, have it switch over again, and analyze the lanthanum. And so it's not going to be great quantitatively because we're losing mass here that we can't quantify. But qualitatively, maybe we can examine the fact that some of these particles have multi-elements present and some only have one element present. And so we were able to show that kind of, and it wasn't perfect, but we were able to show that maybe some of these particles had both cerium and lanthanum present, maybe some only had cerium present, but it wasn't ideal. And really it comes down to just the configuration of the instruments. We really need a totally different instrument to try and examine these different particle populations. And so this is the work I ended up doing in Vienna, where we turned to some new emerging techniques, specifically single particle ICP-TOF, which stands for time of flight, mass spectrometry. And so what that looks like is instead of now having a quadrupole, which acts as a gatekeeper, we now have this time of flight mass analyzer, which works on a completely different principle than quadrupole does. So your particle still comes in and gets ionized by a plasma, but then all of the ions that comprise that particle flow through the instrument to the top extraction region. So there's no gatekeeping involved. And what happens is that in this top extraction region, your cloud of ions get pulsed with the same energy. And if you remember your physics class, Ke equals 1/2 mv squared, right? So if everything has the same energy, and now they're arriving at the detector at different times based off their mass, we can use the time they reach the detector as a way to quantify the mass of the ion. And so all the ions are being pulsed with the same energy. The lighter ions will reach the detector first. The heavier ions will reach the detector last. And so we can get the entire atomic mass spectrum for every pulse that this TOF is producing. And so it's producing a pulse of energy every 46 microseconds. So we get really high resolution data of the entire atomic mass spectrum, which looks like this. And so again, every 46 microseconds, we're getting a whole mass spectrum all at once. And so here, this is just a standard, but what you can see is instead of just getting that one element, now we're getting ruthenium, we're getting indium, we're getting bismuth, we're getting lead, and we can start to see how we might be able to use this information to look at those elemental ratios that we're interested in on a particle by particle basis. And so the kinds of questions we can start answering with this technology are pretty vast. We can start thinking about the isotopic distribution within elements. We can start thinking about core shell materials, delineating between naturally occurring and engineered nanoparticles, starting to get at that question of heteroaggregation. There's just so many questions we can start answering now that we can quantify the total mass range on a single particle basis. And so just a couple of quick examples of how that looks on the most basic side. First thing we started off looking at was just the isotopic ratios within a given element. So silver tends to break down into two isotopes naturally, silver 107 and silver 109. And it typically occurs in a 50-50 ratio thereabouts. Platinum similarly breaks down into about 4 isotopes that we can quantify. And so we can start to look at platinum nanoparticles and silver nanoparticles and look at the isotopic abundance of these different isotopes on a particle by particle basis. And so if we zoom in on this information here, we can see that for our silver nanoparticles, we're getting both our 109 signal and our 107 signal. And it's happening in about a 50-50 ratio. And so sure enough, we're getting about equal amounts of both isotopes. Similarly with platinum, you can see that we get a breakdown of the various isotopes. And again, with enough counting statistics, this ends up breaking down into about the ratios that we would expect. You'll notice though that as we go down in size, we start to run into some issues because we might not have enough mass of each isotope to be detected. And so I'm just throwing this out there as one of the potential red flags that we need to keep in mind when we start thinking about how to look at this data. Beyond the isotopic and bimetallic analysis, we can start again to think about how we delineate between natural nanoparticles and engineered nanoparticles. And so this was some work by my colleague, Antonia Pretorius, where she looked at the concentration of engineered cerium dioxide in the presence of some natural clays. And so you can see that now our natural clays are getting both the cerium and lanthanum, and our engineered particles are seeing only cerium being present. And then we can start thinking about how we might apply this to environmental problems. And so here I had some soil from a mine pit from Macedonia sent to me. And so I took this soil and I redispersed all the little nanomaterials and analyzed it by the ICP-TOF. And what we end up seeing is we can see both the iron spikes and the arsenic spikes present. And so we're trying to demonstrate that the iron is a potential carrier of arsenic, which has implications for how arsenic might be transported in the environment. Lastly, beyond all these interesting questions, we can start turning our focus back to maybe the more natural nanogeochemical environment. And so we already know a lot about geology, but we've never had the capability to study it with a resolution of a single particle at a time. And so we're using this technique now to look at just clays and look at the elemental ratios within clays. And so we can take something like kaolinite, which is this aluminosilicate, having both silicon and aluminum. And if we look at the signal here, we can get some nice aluminum signals and some overlapping silicon signals. And in a mixture of these clays with quartz, which is just silicon dioxide, you can see we get some signals that are just silicon and some signals that are the aluminosilicates. So we can really start examining some really interesting fundamental research questions using this technology. And so for the last part of this talk, what I'm going to go through is a couple case studies in how we might apply this technique to study the environment. And so one case study here, we're looking at some samples collected from the Athabasca River in Alberta, Canada. And my colleagues up at the University of Alberta went ahead and collected water samples from some very DOC, dissolved organic carbon rich tributaries, and then also from the main stem of the Athabasca River. And so we can start to examine how the different chemistries between these tributaries and the main stem of this river play a role in the nanogeochemical environment. And so just to give you an idea of what this site looks like, here we have the Athabasca, flow is going this way. We have a couple of tributaries. We have one down here at the Horse River. We have one here at the Clearwater River. And then we have one here at Steep Bank. And these are all feeding into the Athabasca shown here. And then along the Athabasca, my colleague collected transects along the river. So if we zoom in here, collected both on the west bank in the middle and in the east, and also collected at depth, so near the bottom of the surface, as well as at the top of the surface. And so we're going to look at some of examples of the kind of information that we can produce out of this. Well, the data looks pretty complicated, and so you can see we get a lot of different signals out of here, but if we break this down, we can sort of take out all those overlapping signals and spread them out, and you see that we have a lot of aluminum and silicon and titanium, and if we zoom in on the iron, we can see we have iron with maybe a little bit of lead or thorium or uranium being transported on it. And if we take those signals again and zoom in, you can see we can get some iron and aluminum spikes, we can get some titanium, we can get some very complex multi-element particles as well. And so one thing we can do is we can look at some of the clay geochemistry on a single particle basis. And so this is an example of how we try to present that data, where each of these little dots represents a single particle that we've quantified using this technique. And so what we can do is here we have the iron molar ratio, we have the silicon molar ratio, and we have the aluminum molar ratio. And along these vertices, we have where we only have iron only, aluminum only, and silicon only particle signals. And then along these edges, we have particles that contain only iron and aluminum, those that contain aluminum and silicon, and those that contain iron and silicon. And then within the triangle is where we have our tri-element particles, so iron, silicon, and aluminum. And so we can start to do some interesting things, like we can actually generate a distribution of what those iron to aluminum ratios look like, or what those aluminum to silicon ratios look like. But again, this is being done on a particle by particle basis, so with very, very high resolution. And so what are some of the interesting things that we can generate out of this? We can generate these sort of number proportions of how these different elemental combinations change as a function of where they are in the river. And so some interesting things to pull out of this is we can see that some of our tributaries, like the Horse River, the Clearwater, and the Steep Bank, tend to have higher proportions of iron-only particles. And then if we look at the main stem of the Athabasca, we can see that there's a greater proportion of maybe these multi-element particles, particularly when we take into account things like the amount of total suspended solids. Some other things that we can pull out of this is we can see sort of the change in the size, where here we have aluminum sizes generated here along with silicon, and then our aluminosilicate sizes, and then these tri-element sizes. We see that for these tri-element particles, they tend to be pretty massive. Now, maybe that's an artifact of the fact that we need all three elements present in order to have detectable amounts of these various elements. But that's just something that we're still trying to figure out right now. On the trace contaminant side of things, when it comes to the Athabasca, we know that lead can be transported in surface and groundwaters, both by complexing natural organic matter as well as transporting on the surface of lead colloids. And so here you can see we have an example where we have lead complexing both inner sphere and outer sphere with the surface of this particle. And so if we examine the distribution of iron and lead, we can see that we have a lot of lead being absorbed not only onto the iron particles, but also onto the manganese particles. And so here we have a graph showing the tributaries shown here with the red boxes. And we see that in a lot of cases, there's very little lead transport. And if we compare that with the main stem of the Athabasca, we have a lot of lead not only being absorbed to iron and then our manganese and iron, but also we have a lot of lead-only particles. And so we're trying to examine what that means. And again, we can look at the sizes of these particles, where here we have the total range of iron nanoparticle sizes. And we can see that the iron that's carrying lead tends to be a little bit larger. The iron that has both lead and manganese, larger still. Again, whether or not this is an artifact based off of the sensitivity of the instrument still needs to be examined. And lastly, we can start to look at some trace element geochemistry. So here we have some selected particle signals. You can see we're getting our cerium and lanthanum signal. In some cases, we're getting cerium with all three lead isotopes that we try to quantify. And then sometimes we have these lead-only particles that we try to examine as well. And so by being able to examine these various elemental and isotopic ratios, we can start to produce graphs like this, where we look at, again, on a single particle basis, where each of these points represents a particle. We can look at the relation of the 208 isotope of lead to 207 and 206 and start to develop these trend lines that are actually pretty close to what we would expect in the natural environment. Similarly, we can look at cerium and lanthanum. Here, the red dots represent the information you would get from your total ICP-MS measurement, and the blue dots represent each individual particle where we try to examine cerium and lanthanum. And again, you can see that we get maybe a potential trend, but there is a lot of scatter. And so there's some questions that we can start thinking about examining the enrichment or depletion of certain isotopes or elements on a single particle basis. Beyond the work on the Athabasca, I also have colleagues at the University of Montana, Ben Coleman and Caitlin Perkins, and they collected samples along the Clark Fork River, where here we have Warm Springs Creek, and we're going up here, and you can see we get some nice hydrologic data, the discharge as shown here, as well as some water quality data, both in the conductance as well as pH and temperature. And again, what we can examine is the distribution of iron, manganese, iron with manganese, and various other single elements, multi-element particles. And we can see that, for instance, here we have this drop off in the discharge. We also see a similar drop off in the number. If we look at particle load, we can see that rises commensurately with discharge. And again, we're not just limited to iron and manganese. We can do this kind of analysis with all of the various elements that we try to quantify. And so we can get mass concentrations and particle load concentrations that are, again, based off of this single particle information that we ended up acquiring from the ICP TOF. And lastly, this is just an example of some work that was recently published looking at the South Platte Watershed in Denver, Colorado. And again, I'm just kind of reacquainting you with the area. Here's Golden. And what we end up seeing is that we can start to look at this nanoparticulate zinc in this watershed. And so some examples here, we can start to identify the particle number concentration of zinc. We can look at the average mass of zinc in each individual particle. And we can use this to sort of build maps like this, where now we can start to delineate between our dissolved fraction of zinc, particulate fractions of zinc, and then what we call colloidal fractions of zinc. So maybe those larger than our 100 nanometers. And so we can start to look at how our various maybe pristine, in quotes, systems look like in comparison to maybe our more urban centers around this wastewater treatment plant. And so we can use not only conventional ICP-MS, but we can also use our ICP-TOF-MS to examine this, and we can start looking at how many of these particles detected are zinc only, how many are bound with other elements, and what does this mean for the transport of zinc within the system. And then sort of our last example that I'm going to show you is, what about microplastics? So microplastics are kind of the contaminant du jour. You hear about a lot in the news. They're really difficult to detect in the environment. And part of that's because as we get smaller and smaller for our plastic products, we really start running out of options in terms of how to detect and quantify them. And so right now, the state of the art has us down to about 10 microns in terms of detecting and quantifying microplastic concentrations in environmental samples. So how can we start to remedy this problem? Well, one way is kind of cheating, but it's the way that we're doing now, where we can put metal nanoparticles inside of plastics, and we can start using these plastics as tracers for how plastics distribute in the environment. And so what the environment sees is going to be the plastic. That's going to be what it interacts with. But what we can trace is going to be the gold nanoparticles. And so what this looks like is now we can send our nanoparticles through our ICP-MS, and we can get these various spikes of gold that go along as proxies for when a microplastic was detected. And so we can use this to build up a number distribution of the amount of microplastics in a sample. And so just an example of how this might be applied, we collected some sediment from the U.S.A River estuary, and you can see that what we did was equilibrate some of this nanoplastic with the sediment and start trying to look at how much of the microplastics stayed in the sediment versus stayed free floating versus what we call loosely bound, where maybe we shook it up and try to dislodge some microplastics. And so we can start to examine the various number concentrations associated with this partitioning behavior. And so that was a lot of information, but I hope we all learned something. And so for the future directions, where we hope to take this is keep trying to take all these methods that we developed for engineered nanomaterial analysis and start turning the microscope back to the natural environment and what's already happening there. One of the catches to all this is that we might need more sophisticated methods of data treatment. And so this is an opportunity for things like machine learning. And then the potential to further empower these techniques using some orthogonal combinations with other nanoanalysis techniques to really extend the utility of these various characterization tools. And so with that, I'd really like to thank a lot of my acknowledgements. Dr. Frank Vondekammer, Sarah L. Yubi, and Jan Sherman at the University of Vienna, Dr. Ben Coleman and Caitlin Perkins at the University of Montana have been fantastic to work with. Here at Western, we have a whole host of student researchers, both in collaboration with myself, Dr. Ryder, and Professor Emery, who are all working with these functionalized microplastics. And then for the Athabasca, collaborators at the Colorado School of Mines with Professor Jim Ranville, at the University of Alberta with Dr. Chad Kuss and Professor Bill Shattuck. And then a special shout out to my undergraduate researcher, Haley Holliday, who has been staring at a ton of data for the past few months, helping us work through this extensive data set. And with that, I hope you all enjoyed the talk, and I'm happy to take any questions.
Fantastic. Thank you. There's a lot of chemistry and information there. One of the questions that came in was the correlation, is there one? How do you know there's one between, is there one between microplastics and the larger plastics in the environment?
So how do we know there's a correlation between, so I think I understand this question. I will say right off the bat though, that we're still, as a field, trying to understand the kind of kinetics and mechanisms of polymer degradation in the environment. And so how exactly the larger plastic debris goes from being, a plastic water bottle down to these sort of polyester, polyethylene, or poly PETF, or what have you, all these little microplastics. That's still an active area of research. But larger plastics, really everything breaks down in the environment, except for microplastics, which is part of the issue. And so just the natural weathering processes can break down our larger plastics, and then those generate smaller and smaller plastics through a variety of photolysis and hydrolysis mechanisms.
Okay. So another question came in terms of the risks of engineered nanoparticles and how big of a, unintended consequences obviously is the topic. How do we know how big of a fire we're playing with here?
Well, as with everything, it depends on how, first of all, what kind of nanomaterial, because that's going to determine it. And then what's its potential for entry into the environment is going to be its other kind of question mark. And then what's the relative rate of exposure that we're going to end up seeing. And so I would say, again, I hope it's not a cop-out, but it's an active area of research. And so we can see in maybe laboratory settings that some of these materials have toxic potential, but whether or not that toxic potential remains upon entry into the environment is still very, very much an active area that people are examining still.
Okay. Are there mixtures of materials due to natural processes or from the man-made alloys? So I guess that's an interaction.
Oh, so I imagine, and correct me if I'm wrong here, Wayne, that we're discussing sort of the mixture of nanomaterial populations. Well, regardless, so the mixture of elements for natural nanomaterials just come from the fact that the buildup of geological materials requires all these various aluminosilicates coming together in kind of complex geological formations. And so when they break down, they contain a whole bunch of elements, and some of those elements in the crystal structure of these minerals can be replaced through time. And so we can end up with more complex structures as a result. As far as engineered nanomaterials, there's definitely potential to have multi-element engineered nanoparticles that are generated for a variety of different purposes. And yeah.
Can you speak to limitations of the study of nanoparticles and single particle techniques and some of the breakdown you've been doing there?
Yeah, I mean, the big issue is always going to be it's a whole host of things, but sensitivity is always going to be a problem. So when we talk about nanomaterials, a 2 nanometer nanomaterial only has about 1,000 atoms in it, depending on the D spacing of the atoms in the crystal lattice. And so trying to detect only 1,000 atoms is a really, really, really tall order. And so sensitivity is always going to be a limiting factor. We've seen that this complex mixture in the environment is going to be a factor as well. And then just the fact that we have so many overlapping potential interference present in the environment are also going to be an issue.
Yeah.
And then throughput, I guess I would say real quick, is not only like we could sit there and spend hours on an electron microscope trying to quantify each individual nanoparticle with the resolution we need, but you're never going to get the counting statistics that would be enough to make that a useful kind of quantification.
Right. Are there opportunities for students, either classes or research, to get their hands on the tools you're using to see this stuff for themselves or to get involved?
Yeah, so I'm actually going to be teaching a class next quarter, a first-time temporary course, Environmental Sampling and Analysis, where it's a bit more broad, where we'll be studying more kind of the various tools and techniques out there to sample the environment and make these kind of quantifications. There's always opportunity in my lab for some research. I have a lot of various undergrads who do nanomaterial analysis. And in fact, over the summer, I had a student in my lab doing a lot of nano fungicide analysis. And so that was in Western Today, I think last week, actually, if anyone wants to read up on it.
All right, great. And then maybe a final question. What's next? Where does the research go from here? Bigger or smaller?
Well, I shouldn't say always smaller, but I'm only ever interested in going smaller. I think one of the big questions that we have is how to start dealing with these microplastics. Again, as I showed in that kind of cartoon, we really don't have a lot of options at the moment. And so either by trying to understand their chemistry through using these tracers, or maybe isotopically labeling the polymer itself with like a C13 kind of polymeric structure, and then using that as a way to delineate between other carbon-12 material in the environment. One other thing I'm particularly interested in is, here we've been sending nanoparticles through a plasma. What would happen if we start sending individual cells through a plasma, be they bacteria or algae, and start looking at the intrinsic metal content on a cell-by-cell basis? And what kind of information could we start to glean by developing a single cell ICP-MS?
All right. Yeah, it just gets smaller and better and smaller as you go. Endless job security there. Okay, Dr. Manuel Montano, thank you very much. Next week, we've got Gigi Berardi from the environmental studies side of Huxley College, speaking on human ecology, food-wise agroecology. So a slightly different scale of research looking into food. Again, thank you very much, and good day.